Hello friends, we are discussing packaging of DNA into chromosome. In first part of this topic, we discuss overview of different levels of packaging of DNA into chromosome. We also discuss about histone proteins, non-histone proteins and also function of H1 histone protein separately. In this video, we are going to discuss about packaging of DNA into chromosome in detail. We will discuss about its different levels of packaging in detail. So we already know that in eukaryotes, DNA is organized into chromosome. A highly organized packaging system is needed to fit a DNA molecule into its chromosome. This leads to different levels of packaging of DNA. We are going to discuss these levels of packaging of DNA into detail. So let us see them one by one. First level of packaging, which results in the formation of nucleosome. Nucleosome was described by R. Kornberg in 1974. It is the simplest, basic, structural and fundamental unit of chromatin. Proteins that are responsible for this organization are histone proteins. Four different types of the histone proteins, H3, H4, H2A and H2B, are involved in the formation of nucleosome, that is octamer. So the histones are assembled as octamer. First of all, H3 and H4 histone protein form heterodimer. Two heterodimer then make a tetramer. This tetramer is flanked by two heterodimers of H2A and H2B, and this create histone octamer, that is nucleosome. So this is one heterodimer of H3H4 and this is the second heterodimer of H3 and H4. These two heterodimers then form a tetramer. This tetramer is then flanked by two heterodimers of H2A and H2B histone, and this results in histone octamer, also known as nucleosome. Now wrapping of DNA on nucleosome. Nucleosome is wrapped or surrounded by DNA of 146 base pair length. This coiling of DNA around nucleosome is left-handed coil and it wraps nucleosome 1.67 times. Therefore, DNA is compacted by 6 folds and nucleosome resemble as beads on a DNA strand. Hence, chromatin at this level is referred as beads on a string or thread. So this is the histone octamer or nucleosome, and 146 base pair of DNA surrounds it, or it is wrapped by DNA of 146 base pair length. So this is the DNA double helix which has the width of 2 nanometer. When the histone proteins organize as octamer and DNA is wrapped around it, it has the width of 11 nanometer and it appears as beads on a string. These beads are actually nucleosomes and string is the DNA. Now the second level of packaging of DNA, it is known as 30 nanometer chromatin fiber. It is brought about by H1 histone. H1 histone binds to linker DNA at one end of nucleosome and to the middle of DNA segment that is around nucleosome core. So nucleosome along with H1 histone is known as chromatosome. Hence, H1 histone is not the part of nucleosome core. H1 histone we have discussed in the previous video also. It functions as a clamp. It restricts the DNA into its position. Each chromatosome encloses 167 base pair of DNA, 146 or 147 base pair that is wrapped around nucleosome and 20 base pair to which H1 histone binds or that binds with H1 histone protein. This is the nucleosome core, which is an octamer of 4 different types of histones: H3, H4, H2A and H2B. DNA is wrapped around this nucleosome core. H1 histone protein is not the part of nucleosome, but it binds to linker DNA at one end of nucleosome and to the middle of the DNA segment around nucleosome core. Nucleosome plus H1 histone protein is known as chromatosome. Nucleosomes or chromatosomes are present at regular intervals. Strands of DNA between them, that is nucleosomes or chromatosomes, are known as linker DNA. Nucleosomes compact into a structure having 30 nanometer diameter. Hence, it is known as 30 nanometer fiber. There are two possible models of compaction at this level. First is the solenoid model. Linear DNA with nucleosomes are coiled into left-handed helix. 6 nucleosomes are present per turn. There is central cavity that is stabilized by linker DNA. Helix model, this arrangement is in a zigzag ribbon that twists or supercoils. Solenoid model is the most popular or most accepted model. It compacts DNA 40 folds or 40 times. So what we have seen that nucleosomes or chromatosomes are present at regular intervals, and the DNA between the two nucleosomes or chromatosomes are known as linker DNA. Linear DNA along with nucleosome, which is known as the beads on a string, is coiled into left-handed helix known as solenoid, and the diameter or the width of this structure is 30 nanometer. This is known as 30 nanometer chromatin fiber. Each turn of the helix consists of 6 nucleosomes, or there are 6 nucleosomes per turn of the helix. Next is third level of organization, and this is caused by non-histone proteins. 30 nanometer fiber form loops. Each loop is anchored at its base by proteins in the near scaffold. This scaffold is made up of non-histone proteins. This is 30 nanometer chromatin fiber. It forms loop. The length of the loop is 300 nanometer. The base of the loop is anchored to the scaffold. This scaffold is made up of non-histone protein. This non-histone proteins we have already discussed in first part of this video. You can refer it. The link is given in the description box. Each loop has 20 to 100 kilobase pair of DNA. That is, each loop is made up of 20 to 100 kilobase pair of DNA, and length of each loop is 300 nanometer. Number of nucleosomes per loop is 180 to 300. That is, there are 180 to 300 nucleosomes present per loop. DNA has scaffold associated regions that bind to non-histone proteins to form loop. So there is certain stretch of DNA known as scaffold associated regions that binds to the non-histone proteins, which results in the formation of loop. Next is fourth level of organization. 300 nanometer loops are compressed and folded to result a 250 nanometer wide fiber. The 300 nanometer loop is compressed and folded, and it results in 250 nanometer wide fiber. This is 250 nanometer wide fiber. In the fifth level of organization, tight coiling of this 250 nanometer fiber takes place. This yields chromatid of a chromosome having the width of 700 nanometer. And this chromatid is visible in metaphase chromosome. 300 nanometer loops are compressed and folded and gives 250 nanometer wide fiber. Further, 250 nanometer wide fibers are tightly coiled and gives chromatid of a chromosome that is visible in metaphase. Width of this chromatid is 700 nanometer. Also, the entire metaphase chromosome has two chromatids So the total width is 1400 nanometer. This is sixth level of organization. Chromosome visible in metaphase or the metaphase chromosome has two chromatids. Hence, total width is 1400 nanometer. This is the last level of organization of DNA into chromosome. In this way, DNA is packed into chromosome that is present in the nucleus of eukaryotic cells. So what we can conclude that ultimately chromosome that is produced is 10,000 times shorter and 400 times thicker than naked DNA. This is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.